So let's continue. And this part will be about like how to work with some other datasets. So for now, we worked with a single dataset that we loaded from Geo. And so I'll talk a couple bit more about other possibilities. And one particular thing that actually, let's have a poll. So, do you want to finish by 7, or and I should like cut everything? Well, not everything, but some parts, or we can go to like 7:30. Yes, yes, we want to study more and more till then. Okay, and Zoom doesn't count. Okay. So let's talk about the GeneXpression Omnibus database and how to actually work with it. So for now, we just we only use it to to look at particular dataset. So let's try to find some other datasets with it. And for this, let's go to this Geo datasets portal. NCBI Geo. Yes, the presentation is different one. So the slides is also in the same in the day3 folder, but the second presentation. So yeah, and again, right now there are like 150,000 gene sets, gene data person data sets there, and so in the main website there is a search field. So let's put something into it. For example, LPS. So we want to, or actually, let's do macrophages LPS to be more specific.
I'm sorry.
Yeah.
In Zoom, we cannot see the screen which you show on the slides on the class.
I'm not sharing the screen properly, right?
You see only summary. Yeah, thanks.
Because yeah, now you can see should see my screen moving, right?
Yeah, thank you.
Yeah.
Yes, sorry.
Yeah. So on this NCBI website, let's put macrophages and LPS into the search, and let's go to this Geo datasets database, not Geo profiles. And you should have this kind of results page. Yes. Yeah. So these are different datasets from Geo database, and you can see that there are some titles, some abstracts. You can see like kind of organism this dataset is for, how many samples is there, like what type of this dataset is, like what type of experiment this dataset has. For example, this one is for RNA sequencing, like gene expression by profiling by high throughput sequencing, means basically RNA sequencing. Well, right now at least, or like cDNA sequencing, how people trying to call it now. And what else? Yeah, and actually, let's try to scroll a bit. Yeah, and actually, this our dataset, our example dataset is not on the top page because there are now much more datasets with LPS. But still, you can see that like these keywords are highlighted, so you can briefly scan to understand if it's what you want or what you don't want to look at. You can, yeah, and sometimes you are can be interested in some like more specific data sets. Well, usually you want to specify an organism, so like human, mouse, or maybe some chicken, for example. Yeah, so let's try to find an chicken macrophages treated with LPS. So you can click on this right menu, for example, like for some of the like organisms at least, and yeah, macrophages from male and female chickens. And on the left, there are some other types of filters. One of the interesting ones are in the study type. So you can, well, clicking on customize will get you a list of potential options. So you just check some that you can be interested in. So now, so we'll be adding expression profiling by high throughput sequencing, so RNA sequencing. So like one of the examples, for example, we would like to find some RNA sequencing data set as opposed to microarrays to see maybe some RNA events like from the, like other forms and or whatever. So we are looking for RNA sequencing data sets. And like if you don't filter anything, you'll see like in the beginning there will be expression profiling by array. So this microarray. So now in the study type, now we can click on this expression profiling by high throughput sequencing, and there are no chicken RNA sequencing for macrophages, sadly. Okay, let's go back to like mouse. For mouse, there are RNA sequencing datasets. Yeah, so we click on high throughput sequencing, and now like every dataset has this experiment profiling by high throughput sequencing highlighted here. Okay, yeah, you can also like filter by well, publication date. I don't know when it can be useful, but what can be also useful is like number of samples. So in additional filters, you can show additional filters. You can add like sample count. For example, you are interested in like high, very diverse dataset. So let's sample count at least like 100 samples. Oops, from 100 to many. Yeah, and there are like big dataset with 100 and like 250 samples, or like 160 samples, and so on. And this is all even like RNA sequencing time course with a lot of different samples, and also like time points is usually important quantifiers, and it's probably a single cell. Sure. Let's open this data set. Yeah. Well, in this particular case, it's there are bulk data. Yeah, there are a lot of samples like replicates with 24-hour time point, 12-hour time points, and like many other time points in the middle, and actually 72 hours. That's a nice data set, and pretty recent one. Okay. Yeah, so this is one way to find the datasets. So just put some keywords and maybe select some specific parameters that you are looking for, and from that you can get the results. So another way, maybe not that common, but still sometimes useful, is the GeoProfiles database, web service. So let's go to the geo profiles. And the geo profiles is a database based on curated number of curated datasets for which the geo curators did the differential expression analysis. And there you can put some gene like ECOD1 and find some microarrays. So this is only for microarrays, sadly, that has this gene differentially expressed. Well, so for example, like we see that our ECOD1 gene, well, first this is like macrophage response to a viral infection. So again, like inflammatory activate macrophages. So this is an activation marker. So that's pretty expected results. I don't know why T cells has this ECOD1 because they shouldn't have it. But okay. Yeah, and this in particular can be useful when you have some weird gene that doesn't have entry on Wikipedia, or like very small entry on Wikipedia. So you found it, like maybe it's one of the few differentially regulated genes or whatever, and you're trying to understand what it can do or in what context it can be, like maybe not important, but at least involved. So you can put these weird genes into this geoprofiles query and get the datasets. And from that, in this particular case, like for example, you didn't know about this ECOD1 gene, but from these results you can get that okay, this gene is involved in inflammatory response because so the many results here are about inflammation. Yeah, one thing about GeoProfiles database, it's that it uses different experiment IDs. So instead of GSE identifiers, there are GDS identifiers. So it's because this GeoProfiles web server, like database, is a curated one. So the GDS collection is a sub-collection of all the like curated sub-collection of geo datasets, like geo experiments in geo. So it's much smaller. But yeah, you can load the GDS IDs into Fantados, or there will be corresponding GSE ID in the page for GDS. So you can all use this as well. Yeah. Yeah. Another one, sometimes useful feature is, well, GeoProfiles allows you to find some datasets based on a single gene. Well, you can try to make a similar idea, and in particular, it was implemented in the GeneQuery website web service. So where you can put not a single gene, but a collection of genes. So, and similarly to how MSecDB works. So when you enter a gene set and it outputs a set of pathways, so you can actually make these pathways based on geo database. So if you run an example here, for example, so this input gene set from the example is the hypoxia genes, and you will see some studies where this gene set, like query gene set, has some interesting, like non-trivial regulation. And in particular, so the input like example is for hypoxia pathway, and well, yeah. So you can see that like the third hit is a hypoxic something, so which makes sense because it was hypoxia in the input. So maybe some of you do know that HIF1 alpha gene, like HIF1 alpha protein, for which 2 years ago a Nobel Prize was, like for discovering which Nobel Prize was awarded. So this gene, this protein regulate hypoxic like response to lack of oxygen. So it's actually also like a nice hit. So this allows you to find some gene expression public data sets that has some intersection with your query. And I think we'll try to yeah.
Yeah, sorry. Am I the only one who cannot open the link? I'm just.
People suggest to try the second link. Which is the second?
Okay, I see. Thank you. Okay, thank you.
It works for me. Really?
Maybe because it is
oh, it's HTTP. Okay. Actually, you should just add HTTPS and it should work. No, it still works for me. Yeah. But yeah, the general idea is more or less the same, that you have some kind of gene set and it outputs other data, like public data sets that has some overlaps with your query set, with your gene set. And I think like we'll try to explain like I guess Max will be talking a bit about application of gene query a bit later in this course. So, but for now, just you can look at it as a ways to discover some gene sets, some gene expression data sets. Yeah, and actually, so people voted for studying till the night, till the end of till the midnight. So let's do the like small exercise. Please put your like LPS activated genes, so that we obtained before, like in Fantaros, and put them into gene query. And let's try to see that what the results will be. Hopefully, it will be something about LPS. Did anyone successfully got some results? So do you see some LPS hits? Like maybe like LPS keyword in the like in the. Yeah. Well, let's. So these are the genes. So let's copy them. Go to this gene query. Select mouse and mouse. So we are looking for mice datasets. And yeah, there will be like some again infection. Well, microglia's again macrophages. Okay, maybe not LPS. But another thing that probably should appear, and you see 5, 3, 9. Well, not in the top hit, but still that exists. The data set that we are looking for is there as well. Like not looking for, but the analyzed is there as well. Okay. Yeah, also there is, so it's still like work in progress, but sometimes useful. So there is another like similar tool that is a bit different how the database was created, like the past the GeneSet database. So the GeneQuery database was created by clustering, and there is a version that was created like GeneQuery that has a version that is based on differential expression. So there you can, let's try to do this. It's not as fast, but it should work. And there the results will be not in terms of just overlap with the data set, but actually, yeah, it worked for me, but with a particular comparison conditions that has this overlap with our query set. So actually the query set is the same with hypoxia, and now we can see that this genes overlap with, for example, this comparison is well, low oxygen versus normal oxygen, which is hypoxic environment. So it's very logical to appear in response to hypoxia pathway in the like, query. Or like the first one is also like hypoxia versus normal condition. Now here, if you enter our LPS signature, let's try to enter. Hopefully there will be some LPS results as well. Yeah, and you see that the first, well, actually, all the results are LPS compared to something. So, and this particular one is, well, gene expression infected. Well, it's also like infection. Still like the first all explicitly LPS treated samples. Okay. Yeah. So this like the couple of ways. Well, aside from looking for publication, how to find some interesting data sets. And there is one problem about both like GeneQuery and GeoProfile web page is that it only contains microarrays. And as you probably know, nowadays microarrays are not that used. Well, they are still used, but not as frequent. And usually people do RNA sequencing instead. And the problem with RNA sequencing and database is that while microarrays uploading is standardized to Geo, and basically you have to upload some raw files and gene expression matrix, and there are not much things you can do differently. However, for RNA sequencing, there are, and as you will learn like later, as particular like next week in particular, there are a lot of different options how to actually like do RNA sequencing. So there are multiple protocols for library preparation, and then there are multiple protocols on how to go from raw data to gene expression levels, how what actually is these levels should be like, should be their counts or like some many other numbers. And people discuss a lot like which are better, which are not better, and so on. So basically right now, and likely like forever, there is protocol to how to properly load gene expression level data into Geo based on RNA sequencing dataset. So people load the raw data, raw sequencing reads, and they're required to upload some kind of processed results, but they are all very different. And this prevents it, so it makes it harder to use automatically. Well, because you have to quantify the results each time. on your own. However, like in recent years, like I guess 5 years or so, a number of tools have been developed that allows to quantify the data, like quantify the, like to go from raw RNA sequencing reads to gene expression values like in very fast manner. And in particular, there is a tool called Callista that probably maybe some of you have heard of, which works very fast. And if you do it in like in the United States, close to NCBI web servers, so you can actually quantify a lot of the samples in a very like in a uniform way, in like very cheap, like using the like Amazon Cloud or Google Cloud environment. So one of the first I know groups that did this projects was this ArchS4 project that basically developed a pipeline, like Callista-based pipeline that analyzed a lot of RNA sequencing samples with a cost of like less than 1 cent per sample. And well, if you have like around like 10 samples in a gene expression data set, so basically 10 cents per experiment, which is like a very small sum of money. And basically using this, like with a couple of $1,000, you can analyze, you can process a lot of samples. And this allows so like result of this was like basically a big table containing all the gene expression values for many samples from Geo database. And well, it gets updated regularly, and thanks to these databases, using it, we support like Fantasos does support working with RNA sequencing from RNA sequencing datasets from Geo. Yeah. So for example, let's open this dataset GC dataset in Geo first. Yeah, so you can see that if you look at the platform here, you can see that this was done using a Illumina sequencing machine. So basically, this means that this was high throughput sequencing, like RNA, and there is should be. Yeah, so the experiment type is expression profiling by high throughput sequencing. So this is RNA sequencing, and it's using, so it's not stored in a standard manner in Geo. So let's still like thanks to ArchS4, we can load this dataset in Fantasos. So let's do this. And yeah, so you can read this, that the dataset is also like BMDMs, also like LPS, well, plus interferon gamma treated. So it's very similar to what we had before. Okay, so let's open this dataset. And well, when you open some unknown dataset, so let's look at some markers of activation. ECOD1, for example, the gene that we have been looking at before. So this gene gets, well, let's don't look at the DI branch. So it gets activated in the control branch of this time course. And you can look up some other genes as well. And well, okay, we see that this more or less corresponds with our understanding of what should be going on. So what should we try to do next to understand about our data set? So yeah, normalize. So the first question is, what? So is it linear scale or log scale data? Like any other opinions? Huh? Well, I would like to, like whatever you do, just say is this like linear or like log scale, and then you'll have a poll. Yeah, we can look at the values. And it seems like linear scale. Okay, why? Yeah, well, basically the numbers are indeed pretty large. So it's indeed linear scale. And it's like for RNA sequencing, like in particular in Callista. So this, so the data set is not pre-processed in any way. So basically we have something that is proportional to mRNA concentration, which is linear scale. So yeah, we should somehow normalize it to make it log scale. And yeah, and then we can use some filtering of the genes and so on. And actually, so can you do a PCA plot of this? So this is like an exercise. Let's do a PCA plot. And while you do the PCA plot, I'll tell a bit about this data set. So this is again macrophage activation. So there are two branches, control and DI, and so this is a small time course of like first 6 hours of macrophage activation. Well, so you see that this ECAD1 is upregulated, and the other thing is this is DI. So DI is a abbreviation for dimethyl atacamide. So this is a molecule that we'll be talking about later in this course. Well, in particular the metabolism part. And so this is basically a really important, like a good molecule, like small molecule that can regulate metabolism, like regulate macrophage activation. So who successfully obtained the PCA plot? Okay, I see one person, two people, and 1/2, 2. Okay, 3, 4, 5, 6. Okay, let's. So, well, you can do the like for the quick view, you can actually do it without any filtering. Just like don't forget to normalize it, like to log scale the data, and also like content analysis will be useful as well. So let the PCA plot and color by condition. Yeah. So see here what you can see that well our replicates are pretty close to each other, which is good. And the next interesting observation that this PC1 actually very well corresponds to this time course. So all this like going from 0 to 6 hours actually like corresponds to going from left to right on this PC1. And actually for PCA plot, you can have an inverse picture. So the direction is not important. So maybe it's mirrored, but the general like so it's either left to right or right to left, but the general structure should be the same. And well, in this particular case, it's not that it's not so we don't have these two groups like DI 0 hours and control 0 hours to be in the same place. So that means that it's like not a perfect situation to work with. So we have our molecule not only influencing some other so our activation, but also like already has some influence on the zero on the control points. So it has to be accounted for in the analysis and interpretation. Okay, but otherwise, like it looks like a pretty nice data set. Yeah, but remember, like to replicate, usually don't want to have two replicates. Okay, PCA plot for you is similar to this, right?
No.
No? No, okay. Let's check how no. Yeah, did you quanta normalized? Well, yeah, small difference, but overall, still you have this same numbers. So did you quantum normalize or just log scaled? Yeah, so actually it's useful. Usually you should quantum normalize the data, or actually somehow normalize, because in RNA sequence in particular, well, not in particular, but you can have different depths of sampling. So again, let's just to reiterate. So this is the original data set. So let's do adjust like 1 plus log 2, and maybe yeah, let's try to plot box plot. Probably it's a little bit too much. Yeah, it's not that useful. Let's do. Actually, let's do differently. So we can, what we can do, we can calculate average, and there will be a lot of genes that have 0 values. So these are the genes that, like as I said before, like for microarrays, you always have some number, well, not non-zero number, but for RNA sequence, because it's sequencing based, there can be zeros. So let's filter the genes that has, well, at least, I don't know, one count, like one on average. And yeah, so now let's no, not tools. Yeah, so we just filtered out all the genes that are not expressed at all. So we ended up with like 14K genes. So you can see that the average levels are very different. So remember the picture for like when we have did this plot for microarray, like for GSC 5. So it was a bit different, but not too much. However, here you have very different, like really different like scales. So it's important. So basically, for RNA sequencing, it depends a lot about some random things, like and the depth of the sequencing is different for different samples. So yeah, you have to do some kind of quantile normalization. Well, maybe not necessarily quantile, but some kind of normalization, making the removing this sequencing depth effect. Okay. Okay. Yeah. However, sadly, not all of the datasets are in ArcGIS 4. in particularly, well, if you have the dataset just uploaded to Geo, like just getting public on Geo, won't be in ArcGIS4 because they do like releases once half a year or so. But as I said before, usually, well, not usually, the people are required to upload some kind of process data to Geo, like for RNA sequencing, like processed expression values. And these results are available in supplementary files. So let's go back to the Geo website on this dataset and scroll down. And you see like the supplementary file, there will be this counts.txt.gz file, which is an archive with counts. And well, while people are required, well, this is like requirement for submission, and the datasets are, these submissions are curated, so people, some Geo curators do look at them. So it's not, even that it's not always the case. So sometimes there are no supplementary files at all. But yeah, but still usually there are some kind of gene expression data there. And so now let's load this file, download this file. And for those of you who have Linux or Mac, or have, I guess, 7-zip. So this is an archive. This is an archive. So and you can try to unpack it, like with gunzip command or whatever. Or actually, you can go to the Google Drive folder, and then there will be this counts.txt file. So you can download it from there. But so just for simplicity right now, but you can actually unpack it. And yep, so I did unpack it. And this is a text file with gene expression values. So like it's again like simple text format. So the rows here correspond to genes. So these are, what kind of identifiers are these? Yeah, so these are ensemble identifiers. So this ends moose is for mus musculus. So this is mice again. And we have samples as columns. And well, these are our gene expression values. So pretty straightforward. And this kind of table or similar kind of table you can obtain like from, so this can be, a file like this can be sent to you from some kind of RNA sequencing core facility, or where you ordered to for the RNA sequencing data. So this is, so more or less this kind of table can come from whatever. So, or like basically your own data can be in this form. So, and now let's try to load it in Fantazos. Well, the set in particular is already in ArcGIS4, but I got tired about replacing each year with a new dataset. So let's imagine that it's not in ArcGIS4. So, and we have to load it manually. So there are two ways to do it. One of the well, simple one that doesn't require anything is just you can load this into Fantazos, but let's do like a bit more complicated way. So it's much easier. So the file here, it contains sample names, but it doesn't have any annotation of what are like control samples, what are treated samples, what are time points, and so on. So it's usually much easier to do it manually, like in a manually. And actually you can, and here you can actually like infer what are the samples. So let's do the following. Let's try to open it in some spreadsheet editor. If you have OpenOffice or LibreOffice, it should be very easy. Just open this GCT file with well, this editor. If you have Excel, if you have, it will get more complicated. So there are two ways. You can open it first in a text file, and open the text file, open the Excel. So like in old Excel, you could import the file from, actually, let me try to do this. So I have this old Windows with old Excel, which works much better than the recent ones, in the sense that it allows you to do stuff. So let's this Excel. Yeah, and it works. On Mac OS, you should also have this Open With menu. However, it won't show an Excel application because it's not standard. So you have actually, I believe, in Windows as well. So no, numbers. It's application. Yeah, and actually, I guess both of you have the standard macOS application and Microsoft Excel in macOS. It still will be non-standard. So you have to. I have to screenshot it at some point. So there should be an option to allow non-standard application, after which you will be allowed to select Excel, and maybe it will work. And if not, and you can try to open this Excel numbers first, and then copy and paste a text file, and it will be probably parsed. So, well, I suggest you, in your own time, to play a bit around with this kind, this kind of things, because, well, while some people I know hate Excel, it's usually very well, not usually, but sometimes it's very convenient to really load the text, load the table, look at it briefly, and so on. So just don't forget that the gene identifiers, like gene names, gene symbols, can be interpreted by Excel as a dates, like calendar dates. So which can screw up something. So better install OpenOffice or LibreOffice. There will be no problems like this there. Okay, but the point is, so I'll just show what kind of you expected to do, and end results is available again in Google Drive, so you can use it the same. So let's add some annotation. So basically, well, I don't want to type all these time points, but at least let's get a treatment annotation. Treatment. So these are control samples, and Control C and Control V. So these are control samples, and these are GI samples. So Control C, Control V. Okay, so we have this now annotated table. So we have some kind of sample annotation there. So let's save it. So you can save it as a text file as well, or sometimes there are some quotes problem and so on. So sometimes it's just easier to save it as an Excel file. And yes. And I guess, I believe, yeah, comes. Let me load. Sorry. Yeah, so now you can open it and it will be this, the ones. And let's name it annotated. Okay. And I load it into Google Drive. Yeah, and you can open this file in Fantasus now. So, well, let's file, open your own file, and select this Excel file. Yeah, and so a window like this will appear. So in this point, it's important to mark the parts of this table. So you like, so there are 3 colors. So one is corresponds to data matrix, one is for column notation, and one is for row notation. So here we have our data matrix or GDP matrix starting from this 923 number, right? So we want to click on it so that the blue parts contain only numbers. So the red part contain our sample annotation, and green part contain row annotation. So in this case, we don't have much annotations for genes, but you could have them as well. So and after that, you can click OK, and you will see this kind of table, which looks similar. However, probably you don't know your genes by ensemble genes IDs. Or maybe at least somebody I did remember some maybe Entrez gene IDs, which but definitely not the ensemble ones. And you usually prefer to look at the genes by gene symbols. So let's do add gene annotation, so gene symbols here. And for this, we'll use this annotation tool. So annotate rows from database. So and the database that we'll be using is this mouse database, and we have this ID column that contains ensemble genes, and we want to add gene symbols. So and click submit. And yeah, gene symbol column should appear. So now we can again look up our favorite like ACOD1 gene and see that it's indeed upregulated in our time course. And actually the results should be similar to one. Well, yeah, to the one that we had before. I mean, it's not perfect correspondence because there are different quantification protocols, but more or less the picture should be the same. Okay. Yeah. So this way you can load your datasets, which you can obtain like from whatever, like maybe like as a supplementary files from publications. And one thing that I would like also to mention here is, and you will be discussing with Kostya later, so that for like while for analyzing RNA, like RNA sequencing datasets, you can use this log2 and content normalization scheme. But this won't be as perfect. So the other way, like the proper way, is to use this DSeq2 package, like R package. So that was specifically developed to work with RNA sequencing data. So it has different statistical model, and it works on the raw counts, not the meaning the like in linear scale, so not log scale data, and it can also be done like using this different expression dseq2. Well, it's kind of experimental, but it works. So, and you can select, for example, control 6 hours and GI 6 hours. Also, it will take a bit more time than Lima, but after a while, yeah, this results will appear. And while, yeah, so in the columns there are more or less the same. So instead of average expression, you have base mean, but it's more or less corresponds to this the same. And actually, what I did wrong? Did I do it wrong? No, it's yeah. So you have the same, more or less similar columns as we had before, but they are named a bit different. So we have this base mean, which kind of corresponds to average expression, but it's again like the numbers here are big. So this because we have a linear scale, so it's a linear scale. Well, log2 full change is exactly the same. Well, the same. So this is log2 full change between the two conditions. Well, stat column is kind of similar to t-statistic, but a bit different statistic. But still, you can use it for ordering the genes. Well, and p-value and p-adjusted is nominal p-value and Benjamin Hochberg adjusted p-value. Okay, and as an exercise, so we won't be doing it here right now. You can do again pathway analysis and try to understand what are the pathways that are changed between these two conditions. And another thing that I think I know about we'll be watching about. So it's an exercise how to. So sometimes you would like. So you found a geo dataset, and you really would like to look at it. However, it's not present in HS4, and there are no like supplementary files. So it's hard to load it, so you have to find some computational environment, run some tools and so on, which is pretty inconvenient. However, there is a website called RunASeq, which allows you to do some analysis in the cloud. So actually, you can specify like GSC ID there, and you'll download the sample, like raw data from NCBI, and quantify it using some protocol similar to Callisto. And then you can load it into Phantados. It's a bit complicated to describe, but it should be relatively straightforward. And let me summarize first, and then if some people are interested in, we can try to go over this exercise here, or maybe later in this course. Yeah. So right now, like after this, today and tomorrow and yesterday, and okay, actually no. This is summary about the last module. So basically now we have some understanding of how to find datasets. So especially in like public datasets, and as they said, and as you have seen, there are like 100,000 datasets already available. So the chances that you don't have to do any experiment, and you can just use some other dataset. However, and there are some ways to find it, to find some interesting datasets to work with, like based on publications, keywords, maybe some particular genes or gene sets, and so on. However, also like with RNA sequencing, it's not that standard. It can require some additional playing around with to make it work, like with Phantados or like any other tool. Yeah, and overall, like all these 2 days, is there some? No, there is no some. So, and just to summarize all these 3 modules. So basically, right now, you can like find you can find some data sets, work with it, like check that it's looks nice, if it's like meaningful and so on. If there is like outliers or whatever, process like pre-process it, remove outliers if necessary. Then you can do differential expression analysis to compare like 2 conditions that you are interested in, like to compare. And then after that, you'll usually have some kind of list of differential expression genes, which are not that useful. And the big question is how to like what to do next with them. And one of the ways is to do this pathway enrichment analysis. And while in our case it was not that interesting, so we have this, or at least for me in this case, because we already knew that we have like some macrophage activation, some inflammatory activation profile, and we saw that again, like all the top pathways are inflammatory pathways, which is kind of we already knew. But usually, no, not usually, but frequently, you don't. So you have some weird process, like knockout, for example, and you don't have. Well, you have some expectation, but you don't really know what the results should be, or like will be. So there, the pathway analysis can get you some unexpected results. So, and in general, if you have, I don't know, cases, scenarios where you have like, well, again, either knockouts or maybe like some disease and you don't know like what happens. So this pathways analysis can tell you something. And particularly like the enricher analysis that allows you to talk about like to hypothesize about transcription factors, or there are some disease relevant pathways, or like small molecules relevant pathways. So they also like gives you some potential ideas of follow-ups, or like in general of some ideas of what could go on in your data set. Yeah, and actually also GeneQuery, and we'll talk a bit later about it, also like allows you to find some similar data set that also can tell you what's going on, like can tell you about your own data set. Okay, and to finish, so yeah, so there are a couple of other tools that are kind of similar to Fantazos in the idea, but so you can try them, but I don't really like them. So I think Fantazos is much better. But for like, because there is some kind of conflict of interest, you can still look at them. Like, so I have to put them here, here. And yeah, and we still like we're actually developing Fantazos. So if you have any feedbacks, like bug reports or whatever, so please don't hesitate and send us. Okay, so I guess that's it for these 2 days. So if some people are still alive, we can try to go over this exercise, or I think like at least right now there is, I think there will be time on Friday in 2 weeks, like last Friday. So I guess there can be some time there, so we can so you can try to work on it on your own, and then maybe if you have some questions, you can ask me. So anyone wants to let me know? Like okay, yeah, good. So it's a bit complicated to describe. So and I hope at some point I'll do the like small tutorial about this. But in general, the steps are here, and Yeah, and in general, so there will be actually. Let me, if I do have this. This, so yeah, it should look at some point. So basically, just enter the GSC ID here, and it will process, like it quantifies them. So there will be this, like after quality check, there will be this results web page where you can go to this quantification page and then download accounts in Excel format. And you'll have this kind of table, which is again our genes, ensemble genes, and our samples and accounts here. So, yeah. Okay, so if there are some questions, I'm still here, but and if not, that's it.